So I guess we can start today. I would like to welcome our speaker, Karen Adelman. So Karen did her bachelor degree in biology in State University of New York at Buffalo. And after that, she moved to France to do her PhD. And after that stay, she came back to the US, to Cornell University, where she did her first postdoc at Joel Baines. And after that, she did the second postdoc with John Liss. She started her lab at NIH in North Carolina, where she became the tenure-track investigator and then senior investigator. And after that, in 2016, she moved to Harvard, where she became the professor of biological chemistry and molecular pharmacology. She's also a member of Gene Regulation Observatory at Broad Institute and MIT and Harvard, and also Ludwig Center at Harvard. Karen is interested in regulation of gene expression, and she did really pioneering work in discovery of the promoter proximal pausing and showing that it's a major regulatory step of transcription. She pioneered a lot of genomics work and also in regulation of elongation and show really very basic seminal regulatory mechanism in this step of transcription. She's also interested in the regulatory roles of chromatin and epitranscriptomic, and she received numerous awards. For example, NIH Directory Award for Excellent Science. And in 2023, she was elected to the American Academy of Arts and Sciences. And in 2024, she became an international EMBO member. I would also like to mention that Karen is really active in organizing the meetings and courses for students, which is very nice, and she's always very helpful. I actually participated in one of these courses 20 years ago. That's how we met, and she was always very helpful. And I think I can also disclose that one of the EMBO nominees described her as very successful and still having a normal life, which I think we really need to hear. And with this, I would like to give Karen the medal and MENDEL medal and the certificate.
I am moved and touched to be here and really, really honored. It's been just an absolutely wonderful day of science, a couple of days of touring around Brno, and the last 2 hours just being here in this abbey and seeing where Mendel did his work is just really, as someone who's done genetics sort of for many, many years, to come back to kind of where it started is just really moving. So thank you so much, and thank you to everyone who's here for being here. And I'm very happy to tell you a bit about the work that's going on in my lab. So thank you. Before I start telling you about transcription regulation, I just want to sort of make a statement about, I guess, career trajectories that's maybe aimed more at the trainees and the younger people in the audience. Because I think maybe I can correct a misconception that I probably had when I was in your place, is that when I would see someone at a certain stage in their career, I sort of had this imagination that they had, maybe started as a high-achieving student, they knew exactly what they wanted to do, where they wanted to go, and then boom, they went straight up and became a Harvard professor. It just worked out all worked out so simply like that. And I have to say that at least from my experience, it was kind of a little bit not like that, and a lot more like I was an undecided student, and I went all over the place. And then I ended up in a place that I think is where I belong. But I just want to say for everyone who's maybe sitting there and saying, yeah, I really like what I'm doing. I like science, but I don't yet know what my calling is, that I think that's normal. And I think that really what this whole path in science is about is finding what you are excited about, what really interests you, what you're passionate about, and then to take that, to grab that with both hands, and then let your sort of your path, wherever it goes, take you to wherever you belong. And I started as an undergraduate thinking I was interested in science, but not really knowing what I wanted to do. And when I fell in love with biology, I thought that meant I would go to medical school, because I didn't know that you could actually have a job doing experiments, right? I didn't know that GregorMendel.com would be like a thing where we could get jobs doing genetics. It's amazing. And I found that out as I was an undergraduate, and I had an opportunity to get into the lab. And I knew right then and there that I could go to quote unquote work, and it didn't feel like work. And that's that moment when you say, haha, this is the thing for me. This thing that you actually love doing, and that you can do, and you just feel like it's something that comes out of yourself. And so I just encourage everybody to explore broadly, so that wherever it is that you end up, that's the place that you're meant to be, and it comes from finding your own passion. So for what that's worth, that's my advice to the trainees. Sort of, if you don't know what you want to do, that's really okay. But find your passion and then follow that, because what we all want to be is good at what we do, but able to still be kind of a normal person. So I'm very honored to be kind of a normal person. All right, so let me then get into the science and sort of start by getting us all on the same page about transcription regulation. And so what I want to sort of remind everyone is that transcription of, let's say, mammalian genes has to initiate at a particular place. That's where the gene sequence begins. And this enzyme, the RNA polymerase, has to find that transcription initiation site, which it does with the help of a bunch of proteins that I'm not gonna bother you with all the acronyms, lots of them, but they help the polymerase find where to start, and that's very much a regulated process in our genomes. But that's not where regulation ends. So when I started my lab, we kind of thought that that was the biggest part of regulation. And if I've contributed something to the field, it's really to say that there's still a lot after that. And I would argue even still now, there's a lot left to learn. Now, what my lab had focused on for many years was that after the polymerase initiates transcription and synthesizes this short RNA of maybe 25 to 60 nucleotides, it's bound by these factors, SPT5 and this negative elongation factor complex, and it holds the polymerase in the promoter region, and it sort of says, hey, wait a second, you're not ready to go just yet. Wait here in the promoter region, and this is called promoter proximal pausing that Dalibor mentioned. And this is really kind of like a checkpoint in transcription. And this paused polymerase is not released into the gene to actually make an RNA until it's visited by this kinase, the positive transcription elongation factor kinase, or CDK9, which Dalibor has studied. And this is what triggers pause release. It gets rid of this negative elongation factor and really creates a docking surface on the polymerase for a bunch of proteins that help the polymerase transcribe productively across the gene. And of course, to deal with nucleosomes, because there are these obstacles, these nucleosomal obstacles, all along the polymerase's path, and it has to have these special proteins that help it transcribe across the very long, oftentimes, transcript. So my lab had studied pausing for many years, and I'll just say that the question we had most often was why? Why would, if once you find the promoter, why not just go? Why do you have this extra step? And so for many years, we hand wave, we kind of make things up, and we say, well, it makes sense to us that. Maybe it's because. And so one thing that we found in early work from my lab in about 2008, 2010, is that having this polymerase sitting here sort of keeps nucleosomes at bay. So once you open up the promoter, once you get rid of nucleosomes from the transcription initiation site, and you start this pausing process, that really prevents new nucleosomes from coming in. So in that sense, you can have a gene that's not fully on, but you also don't return to this really repressed or off state. So it's kind of this metastable state that allows something to be then potentially activated by certain signaling and cues. And indeed, this promoter proximal pausing was really first discovered by my postdoc advisor, John Liss, who had studied this in the context of the heat shock promoters, where the paused polymerase was sitting there waiting for heat shock to release the polymerase into the gene. So this also stimulates additional rounds of transcription. We hear a lot about bursts of transcription, and we know that the scaffold of transcription elongation, or transcription initiation factors can be stabilized by the paused polymerase, and that this allows you to get this nice synchronous burst of gene activity when a signal is received by the cell. So we have a polymerase waiting to go, the signal comes, the polymerase can shoot off into the gene in this fairly synchronous gene activation. But the thing that John and I said in an early review that we wrote, which is really an idea that we really liked, but to be honest, we were just kind of imagining at that time, was that this serves as a checkpoint to make sure that the polymerase is really fully matured, fully undergoes this proper transition to maturation before it goes into the gene and encounters these first nucleosomes. And so this made sense to us, but we really didn't have concrete examples of why this was super important. So what I want to tell you today is that after almost 20 years of having my lab, I can finally say that I think this is true. I wasn't just making this up because it sounds good, and I want to tell you a little bit about why we think this checkpoint is so important and how cells struggle when they don't have proper quality control to make sure that only productive polymerases go into the gene. And I'll just sort of give you a high-level overview of why productive elongation is important. And that's because our genes are super long sometimes. Sometimes they're short, but many times they're tens and tens of kb, 100 kb. And the polymerase, the RNA polymerase, unlike the DNA polymerase, has to go all the way from the start, all the way to the end, or else a full-length, mature RNA is not formed. All right, so DNA polymerases can start over again, but the RNA polymerase has to finish what it starts in order to make a proper RNA. And this is made difficult by what we call cryptic termination sites that are really all across our genes, in particular across our introns. And so I want to just note that our mammalian introns are so big because they've been repetitively invaded by retroelements, by transposons. And those are often AT-rich, and they have sequences that look like a polyadenylation signal. It's just AAU-AA. It's not a lot of information content, and so AT-rich introns often contain these cryptic termination sites that if the polymerase isn't really sort of a fully mature complex, can trick the polymerase into thinking that that's where you stop. And this premature cleavage and polyadenylation can cause real problems that are linked to a number of different human diseases. And in many senses, this is related to the fact that these RNAs contain a bit of an intron. Right? So if you terminate and polyadenylate, when you're in the middle of the intron, you can't splice because you haven't even gotten to the next splice site. So this retained intron causes issues if any of this RNA sequence makes its way out into the cytoplasm. And that's the case because if this sequence is translated, your cells have never seen that peptide before. So these are often viewed by your adult organism as a neoantigen. So we don't want introns to be translated. But furthermore, even if the intron is not translated, I just told you that introns are full of retroelements. And those retroelements can form double-stranded RNA and or other structures that our cells view as viruses. And so you also get some inflammatory or immune surveillance responses, viral mimicry, people often call it, when these retained introns find their way into the cytoplasm. And so this causes, a lot of difficulties, and the cells go to great lengths to prevent this premature cleavage and polyadenylation. All right, so the field and the work of many people had shown us that the polymerase pauses in early elongation, and that this pausing is released by this kinase that really fully licenses the polymerase to go into productive elongation. But what happens, for example, at the heat shock genes, where the polymerase is paused, it's waiting to go, but PTEF-B kinase never comes. The heat shock doesn't, ever arrive, or there's no stress in the cell. And so we don't want this paused polymerase to sneak into the gene. We don't want it to escape into this gene body. So what we've realized in the past maybe 5 to 10 years is that our cells have a completely, sort of distinct complex that's meant to survey the paused polymerase, and in the absence of PTEF-B, target this complex for premature termination. So it really takes that polymerase and it just removes it from the DNA. And it does that using the exact same enzymatic activities that our canonical cleavage and polyadenylation machinery will use, namely a phosphatase to remove any stimulatory phosphorylation, and an endonuclease to cut the RNA and destabilize the elongation complex. Now, this complex is called integrator, and I'll talk to you about this for a minute for those of you who haven't heard of the integrator complex yet. Again, without trying to go into more details than many of you care about, I'll just say that it's a 15 to 20 subunit complex with the canonical integrator subunits labeled INTs 1, 2, 3, 4, 5, to 15. And when this complex was first discovered by Ramin Shikatar, there were really only 2 subunits that we could make much sense of. And that was integrator 11 and integrator 9. And this we understood because these were paralogs of 2 proteins in the canonical cleavage and polyadenylation machinery. So this all arose from gene duplication. So now we have one termination machinery that works at the gene end and another that works at the gene start. So it's kind of cool, but they're actually direct paralogs. And in this, there are 2, it's almost like it's a heterodimer of 2 different, but quite similar structures here with a beta-lactamase, beta-caspase structure, where INTS 11 has the active RNA endonuclease, and INTS 9 has a degraded endonuclease, so it can associate with RNA, but it doesn't actually cleave it. And so we kind of knew that integrator cut RNA, but we didn't really understand how else it was causing termination. Much more recently, my lab and others discovered that protein phosphatase 2A subunits, A and C, are actually integral parts of the integrator complex. So it doesn't bring, it doesn't have its own phosphatase activity, but it brings PP2A with it to dephosphorylate the polymerase. So if any of you are PP2A experts, you know that PP2A, A and B should usually be bound by a PP2A-B subunit, which gives it specificity. In this situation, integrator is the B subunit. And so integrator basically tells the protein phosphatase where to go, and it really just broadly dephosphorylates the transcription machinery. There have been beautiful structures of the integrator complex solved by Patrick Kramer's lab, and this is a really nice one that Isaac Fayanu made for me using my preferred colors for integrator. And what I think you can see here is this is a really sort of modular structure with this sort of tail. And then this, here in this sort of oranges and yellows, this is the endonuclease domain, this is what cleaves the RNA. There's a phosphatase domain, which dephosphorylates the polymerase. And we can see in their structures where this docks on the paused polymerase that it recognizes both the polymerase and SPT5 and this negative elongation factor, which I told you really only binds in the promoter proximal region. And so when it docks on the paused polymerase, it's a stable docking, and that's probably what gives it specificity for this early elongation complex. But when it docks, it puts this in the proper place to dephosphorylate the polymerase, and this in the proper place to cut the RNA. So it's a really beautiful sort of modular structure. Okay, so integrator then, sort of a complex. We don't really understand what most of the subunits do, but human genetics was telling us that integrator was doing something important because nearly all of the subunits are essential proteins. You can't find them sort of absent in alive humans. And the only exception to that is integrator 6, because it has a paralog 6-like. Now, you can't have a complete knockout of the integrator subunits, but you can have mutations within the integrator subunits, and these are actually quite common. And these integrator mutations are often things that cause a deficiency in integrator function. And a good example of this would be a haploinsufficiency, where one of the subunits is mutated and non-functional on, for example, the maternal allele. And so you have some integrator activity, but it's just not up to sort of its full strength. And this is associated, this haploinsufficiency or mutated allele is associated with a number of different diseases. I know this is a horrible graphic. All you're supposed to get out of it is that integrator is important and worth studying, and that there are a lot of mutations in everything from cancer and heart disease to there's a lot of ciliopathies, spermatogenesis, sort of ciliopathy associated. And then there are a lot of neuronal disorders, everything from neurodevelopment to neurodegeneration. And I want to just stop and highlight these neurodegenerative disorders for a moment, because we're going to come back to these at the end of my talk. And so the first cohort of this was found in Holland with a group of patients that all sort of presented with a very complex disorder that was a cerebellar hypoplasia. And so this is mostly cerebral spinal fluid instead of cerebellar neurons. And these patients also suffered from intellectual disability, seizures, motor impairment, and had a number of other sort of traits, including heart defects. And so these patients, which were first described, had mutations in either integrator 8 or integrator 11. More recently, even more severe mutations were found in integrator 11 that actually, unfortunately, killed this young boy when he was only 5 years old. And so what I want to point out, going back to that modular structure, is that mutations in the phosphatase, the endonuclease, or the core domains all lead to very similar disorders, saying that this is really an issue with integrator function that's causing somehow neuronal cell death or failure of neuronal migration. So we wanted to understand how integrator deficiency contributes to disease. And so to first do that, we wanted to just do some basic molecular biology of the sort that my lab knows how to do. And I'm just going to walk you through sort of the history of where we were up until the sort of more current story that I'll really focus on. So my lab studies both Drosophila and mouse and human systems. And we started studying integrator in collaboration with Eric Wagner working in Drosophila. And one of the things that we had done in this early work was to deplete integrator 8, which is part of this shoulder that associates with the phosphatase. And we depleted this using RNA interference in flies, and we did RNA-seq, very simple experiment. And what I want you to really get from this is that lots of genes are upregulated, not too many genes are downregulated, and that this was consistent with what we were thinking of as a termination complex, and it really suggested that many of these genes that are upregulated, those are the genes that integrator is normally working at to attenuate their activity by performing this quality control role at the promoter. So we see lots of gene upregulation. And what was interesting is that it wasn't just the heat shock genes that showed this upregulation, these things that were known to have this paused PAL2 that wasn't visited by PTFB, but a number of stimulus-responsive pathways associated with immunity, oxidative stress, et cetera. So, this suggested that integrator was attenuating the expression of these genes. We wanted to get a really good handle on what that meant, sort of molecularly. So we performed an assay called precision run-on sequencing, that was first developed in my postdoc lab. And this is a nice way of taking a snapshot of exactly where the polymerase is on the genome at any moment. And the way this works is you have your happy growing cells, you take them and you rapidly put them in a cold buffer with a little bit of detergent. It pokes holes in the cells, NTPs flow out, everything kind of stops, the polymerase gets trapped in place, and you freeze it. When you're ready to do your experiment, you warm everything up, and you replace the NTPs that flowed out with biotinylated NTPs. So now a polymerase that's engaged and still on the DNA is very helpful to you because it adds one biotinylated NTP to the growing RNA chain. So it happily biotinylates your RNA for you. And it only does that if it's already engaged in transcription again, because this has been washed with detergent that prevents new initiation. So now you have a little biotinylated RNA, you can fragment it, you can pull out the biotinylated part, you put ligate adapters on, and then you sequence from this 3' end with the nucleotide right next to the biotin telling you exactly where the polymerase was sitting when you harvested your cells. All right, so it's a very nice mapping of polymerase. And so, we'd use techniques like this and others to really just document this phenomenon of pausing, and that we can use this to measure, in a sense, the residence time of polymerase on the genome. And here I'm just showing you an example of polymerase at the GAPDH gene, where you can see this pause, this slow-moving polymerase in the promoter proximal region. Then it gets licensed, so it now moves quickly as it goes across the gene body. And then after termination, it's also dephosphorylated, and these positive factors are sort of kicked off the polymerase. And the polymerase then starts going more slowly, and so you can see this very different kind of pausing at the gene 3' end. So ProSeq is a really nice mechanism or a nice technique to give you some dynamics of the polymerase as well as its localization. So what we did then is we did ProSeq in these Drosophila cells where we looked at all of the integrator target genes and we asked, is the polymerase even there? And where is it? And this little arrow shows you where the promoter is, and this is all of the integrator target genes. And I think what you can see is that the polymerase can get to these genes, it can start transcribing, but it doesn't make it very far away from the promoter. But when you deplete integrator, this integrator 8 subunit, now you see that these polymerases can be released into the gene body. And so this was really our first concrete example that integrator was binding to these paused polymerases and keeping them from entering the genes. And when we got rid of integrator, polymerases, immature polymerases, we imagine, were inadvertently escaping into the gene. And so what kind of genes were those? I already told you that in the Drosophila system, they were a lot of stress-responsive genes. We've since done very similar experiments in the human system, so this is just 293 T cells, again with integrator 8 RNAi. And again, we see immune signaling, things having to do with DNA damage, and oxidative stress or hypoxia. Now we've moved from siRNAs into things like degrons, where you can have targeted protein degradation in just a matter of a couple of hours. We can do the same kinds of experiments now using ProSeq or transient transcriptome sequencing. And again, we see here, this is mouse embryonic stem cells, 4 hours degradation, the same kind of genes upregulated. So it really didn't matter which subunit you were knocking out or which system you were in, from flies to human, siRNA, whatever. We were always seeing, and we, now meaning really pretty much everyone in the field, was seeing that the genes that were upregulated in the absence of integrator were stress-responsive genes. And so that really led us to a model where when you had paused polymerase and PTEF-B, in the absence of stress, for example, didn't get sent to that gene to turn that gene on, integrator went to that gene to attenuate the expression of these stress-responsive genes under normal growth conditions so that they're only turned on in the conditions where you want those stress-responsive genes to be activated. So this is a really nice way of keeping stress-responsive genes poised for activation without having leaky expression. All right? So this is the model in the field. Very happy with ourselves. You will probably still notice that I haven't answered my question, which is how integrator deficiency actually contributes to disease. And so this was the work, what I'll describe now is the work of Apurva Balapuri in the lab, an incredibly talented postdoc, and this work for people who want to see the 5 gazillion controlled experiments that the reviewers kindly asked us to perform. It's here in the supplement of this manuscript. All right, so what Apurva wanted to do was really make sure he was studying integrator proper, the whole complex. And so he, in collaboration with Kai in Eric Wagner's lab, made use of this cell line that Kai had generated, where there was a degron tag, an auxin-inducible degron on integrator 1. So this is the core that really is essential for anchoring integrator to the polymerase itself. So if we get rid of this core, we would imagine all of integrator would come off the polymerase. And this is just showing you that we have good tagging of INS1 in our parental, tag line, and we add auxin and the protein is gone, but it's specific to INS1. These other subunits are not rapidly degraded, although, as I'll show you in a moment, they do indeed come off the elongation complex. So just to sort of validate the system, Apoorva wanted to ask whether or not integrator 1 loss in this 293 T cell system activated the kinds of genes we expected. So he did transient transcriptome sequencing, and this is really just another way of looking at newly synthesized RNA, where you have your cells, they're happy, they're growing, and we, 4 hours after adding auxin or not, we add 4-thiol-U to the cells for 10 minutes, and then we harvest the RNA, we fragment it, and then we use this 4-thiol-U as a handle to purify just the RNA that was made in that 10-minute pulse. And so here we have, we can then have a nice measure of what RNA was just made in the last 10 minutes, and we can do differential gene expression analysis, and we can see that, as we'd seen in many other systems, lots of genes are upregulated. There are many of the same stress-inducible and immediate early genes that we and others had described, things like FOS. And this is the TT-seq in the minus auxin or the integrator depleted conditions, and these genes are coming on very strongly. When we look at the kinds of genes, we, as you probably are anticipating, see a lot of genes involved in inflammatory, reactive oxygen, and DNA damage responses. And so stress-responsive genes are coming up in this system, as we might expect. One of the things, though, that we noted in our mouse system, also with the Degron, that I think is important to note here, is that the genes that were upregulated were not only stress-responsive genes, but they were also very short. And we kind of reasoned, again, maybe a little hand-wavy at that point, that maybe this makes sense because if the polymerase, the immature polymerase, is escaping into the gene, it can make it all the way across a very short gene, but maybe it has a hard time making it all the way across longer genes. So those genes wouldn't be fully upregulated. And indeed, what I think you can notice here is that the downregulated genes are in fact much longer than the unaffected or the upregulated genes. And the converse is true as well. Long genes are downregulated and the downregulated genes are long. And so this sort of gave us this idea that the integrator, in integrator's absence, the polymerase that kind of escapes or sneaks into the gene isn't fully fit to make it all the way across our canonical long mammalian genes. So I always use the analogy that, in the absence of integrator, it would be like if somebody let me run the Boston Marathon. Not a good idea. I could make it maybe 5K, maybe 7K, maybe not, but I certainly couldn't make the whole marathon. And so I'm kind of like these kind of crappy polymerases in the absence of integrator, trying to do something that's maybe I'm not quite up for. Okay, so what does this do then, all this nascent RNA formation, what does this do to the steady state RNA? Because phenotypes are actually going to be caused by steady state RNA. What's the RNA that's out there being translated? So we wanted to address that, looking either 4 hours or 12 hours after integrator depletion, then with auxin. And so we did RNA sequencing. We kind of clustered any of the genes that were changed at either of these time points. And here's just this clustering. And we have sort of 5 distinct clusters here. I'll just call your attention to this first group, which is transiently upregulated and then back down. These are the genes that we had studied before, many immediate early genes that are turned on and then turned back off. And these are genes like Jun and FOS. Then we look at these genes that get turned on early and stay on. And now we start to see gene ontology categories appearing. And these, again, are stress-responsive categories. But interestingly, when we look at the transcription factors whose binding sites and motifs, et cetera, are correlated with the expression of these genes, we see, in fact, the transcription factors, Jun and FOS, being the top most correlated transcription factors here. And so this then tells us that perhaps the transcription factors that are turned on really early start to play a role in shaping or sculpting the later transcriptional events that are happening, not immediately, but longer term during this depletion. And this became even more interesting when we looked at this later time point, these genes that we hadn't studied before. They only come on 12 hours after integrator depletion. Now we see really a full repertoire of stress-responsive genes. And when we look at the transcription factors that are involved, FOS and Jun are still here, but now there's a new hit that's really becoming sort of our top hit here, which is this transcription factor called ATF3. Now, motifs are motifs, but we were already kind of interested in ATF3 because of a control experiment that we had done to validate our system. And this was an experiment where we had done quantitative mass spectrometry on the polymerase, and actually on the elongation complex, because here we're pulling down this SPT5 protein. And what we wanted to make sure of was when we added auxin and we lost integrator 1, that the entire integrator complex came off the polymerase, right? Because that was our model, but we wanted to test that model. But what we noticed was that when integrator comes off the polymerase, we actually see a couple of transcription factors now appear enriched in the neighborhood of this elongation complex. And so because we and others had seen Jun as an integrator target before, we weren't that surprised to see Jun now enriched. But even more so enriched was this ATF3 transcription factor. So now motifs plus protein got us kind of interested in this factor. Now, what is ATF3? So ATF3, along with its partner ATF4, is a key part of this integrated stress response. Right? And so this is a super highly conserved, multifactorial stress response that makes use of stress sensors, or these are kinases, that can really read out in the cell anything from proteotoxic stress to nutrient deprivation to double-stranded RNA. And when these sort of problems are sensed, it sort of goes through this whole pathway to try to restore homeostasis, and it reprograms gene expression by both changing transcription and also stopping translation. And so, for example, if the cell thinks it's been invaded by a virus, it wants to stop translation because it doesn't want to make viral proteins while it's turning on the transcripts and then making its own proteins that it would use to be protective. All right, so we inhibit protein synthesis, and then we also activate these stress-responsive genes. Now, what's important is that the integrated stress response is meant to restore homeostasis. If that works, all is good. You keep going forward with your life as a happy cell. But if this doesn't go forward, then this really triggers for the cell apoptosis. And one thing that struck me as I was reading about the integrated stress response was a lot of recent work from Peter Walter, which is summarized really nicely in this review he wrote for Science, where he's finding that a chronic or dysregulated integrated stress response is really playing a large role in a number of human diseases, especially those associated with aging. And things that he was particularly interested in were neurodegeneration, as well as cancer, and a number of metabolic and signaling disorders. And so what struck me at the time was really the overlap, the sort of intriguing overlap between the kinds of diseases that are associated with integrator deficiency and the kinds of diseases that are associated with this chronic activation of a stress response. So we wanted to look into this. And so, a simple question is, does integrator depletion actually turn on the genes that are associated with the integrated stress response? And so indeed, if you look back at our TT-seq, you see that both of the transcription factors that partner with ATF4 are very strongly upregulated within the first 4 hours of integrator depletion. So these are indeed direct targets. Now, when we look at the protein levels, we can see that ATF3 is not only upregulated at the RNA level, but at the protein level. And we can look at ATF4, which is translationally upregulated during the integrated stress response, and we see that this too is triggered by the loss of integrator. So that suggests that there's a there, and there's something to dig into. So we next wanted to ask whether or not we saw any evidence of inhibition of protein synthesis, and we first looked for phosphorylation of this factor EIF2 alpha, and indeed you can see when we add auxin, the cells start phosphorylating EIF2 alpha, suggestive of this protein synthesis or translation inhibition. Now, when the cells inhibit translation, they take a lot of these stalled ribosomes and the sort of messenger RNAs, and they store them into what they call stress granules to kind of protect them and the cell from the cell and the cell from them, for example. And you can study this formation of these stress granules by following this marker G3BP1, which really nucleates the formation of these stress granules. And so what we can see is that in happy cells, G3BP1 is sort of all throughout the cytoplasm, but when we add auxin and we get rid of integrator 1, we see that now over time, you start forming these clear puncta, which is really indicative of stress granule formation. And so this and other experiments that show the cessation of protein synthesis really caused us to appreciate that all the hallmarks of the integrated stress response were then being observed in these integrator-depleted cells. So then the question is, how is it that loss of integrator causes or triggers the integrated stress response? And so here we're talking about a transcription terminator. So there's something here that's kind of calling out and saying, maybe it's double-stranded RNA. So that's where we started. And so we wanted to know whether or not double-stranded RNA was being formed and could accumulate in the absence of integrator. So Apoorva performed indirect immunofluorescence using an antibody called J2, and this recognizes double-stranded RNA in a sequence-independent manner, so it really just reads out any double-stranded RNA longer than about 40 base pairs. And here what I think you can see is that during a time course of auxin treatment, you see accumulation of double-stranded RNA in the cytoplasm, and this is particularly important because double-stranded RNA sensors, including this sensor PKR, are in the cytoplasm. And so it's really the cytoplasmic double-stranded RNA that kind of gets the cells quite freaked out and causes them to really turn on this integrated stress response. And so if PKR, this protein kinase R, is excited, it undergoes autophosphorylation. So Apoorva also looked for autophosphorylation of PKR. And again, you can see that in the absence of integrator, we see this autophosphorylation. Finally, Apoorva wanted to really sort of nail down that double-stranded RNA, here shown by the J2 antibody, was being recognized or within the proximity of PKR. So he did a proximity ligation assay where these yellow dots show you where double-stranded RNA is within 40 nanometers of PKR. And you can see that when we add auxin to these cells, we see lots and lots of dots, which really tells you that the PKR is now seeing double-stranded RNA, that it wasn't in our control or wild-type cells. All right, so where is this double-stranded RNA coming from? What we wanted to do is look at all of the double-stranded RNA, but really particularly to focus on the double-stranded RNA that was being bound by and recognized by PKR to trigger this response. So we immunoprecipitated both kinds of RNA, we sequenced it, and then performed peak calling. And as many of you in the audience can probably predict, the vast majority of our double-stranded RNA peaks mapped to retroelements, to transposons in the genome. And that was true for both flavors of peaks. And so what was kind of interesting, and we'll focus here on the PKR-seq peaks, but the same was true for all double-stranded RNA, is that these were particularly enriched in a kind of retroelement called SINE, and in particular in ALUs. And so these are within the human genome, particularly they're relevant in euchromatin, right? So we have lots of retroelements all over our genomes, and some are the sort that kind of lie near centromeres or tips or in heterochromatic regions, where the chromatin or the DNA methylation keeps those regions very much closed up, and so those are inactive regions. And that was initially what we were thinking, is that you get rid of integrator and those regions kind of become active. But what this is suggesting is that we may now be making transposable element RNAs from active regions. And so because ALUs are enriched in introns, we looked at the distribution of these peaks. And indeed, we see that whereas most of our genome is either intergenic space or inactive genes, most of our peaks were actually coming from the introns, specifically introns of active genes. All right, so what's happening? So I'm going to show you an example gene to kind of give you a sense for what we think is going on here. And so here's just a gene with a very long first intron. And this is our TT-seq at the top. And then these are our J2, our double-stranded RNA peaks, and these are our PKR peaks. And here is just a little region where there are a lot of peaks, and I'm just going to zoom in on that here. Now, what Apoorv has done here, which I think is particularly interesting, is he's shown all of the alus as arrows. And if the alus of basically the same age group, meaning that they would be complementary, have integrated into the genome in an inverted formation, he gives those color green. And so what you can see here is that we're forming double-stranded RNA when complementary alus are located in the gene, or in the introns, very close to one another. So what we think this is telling us is that this isn't two completely separate pieces of RNA coming together to form a long double-stranded RNA, but the RNA is actually folding back on itself through these inverted repeats and forming an intramolecular double-stranded RNA. So the other thing I wanted to point out is that if you look at the transcription by this TT-seq in normal cells, you get this polymerase going into the gene, it enters the intron, and it goes across the intron. In the absence of integrator, in the absence of this quality control machinery, you see much more polymerase escaping into the gene, and it sort of gets so far. But as you notice, you go across this very long intron, less and less and less polymerase is making it each little 10, 20 kb. And by the time you get to this point, there's no more polymerase than you had in the wild type samples. And so this is really what we consider in the field very indicative of premature termination, of polymerases that start but don't finish what they've started. And so this kind of pattern is true not just at this example gene, but if we look at all the genes where we find PKR peaks, when we add auxin and get rid of integrator, we see much more polymerase going into the gene, but actually less making it to the end. And so this really suggests that this immature polymerase that is sort of precociously going into the gene just doesn't quite have what it takes to get it to the end of these genes. And as you may be sort of predicting, these genes tend to be much longer than the average gene. So these are exactly the kinds of genes that would be causing trouble for these immature polymerases. And of course, the longer genes tend to have the longer introns. And so they also have plenty of cryptic polydenylation sites to cause trouble. Okay, so what's our model? Our model then is that in normal cells, integrator sits and waits and sort of probes the paused polymerase and makes sure that the only polymerase that is able to escape into the gene body has been properly visited and phosphorylated by PTEF-B so that this now productive elongation complex can ignore cryptic polydenylation sites, go all the way to the gene end, and just stops and sort of undergoes polyadenylation where it's supposed to. That allows the full intron to be transcribed and then removed. And then it sort of doesn't matter if there are transposable elements or alus or whatever in the intron, as long as it gets removed and degraded in the nucleus, because that's not where the double-stranded sensors are. Then you export a messenger RNA that is functional and can go on and be translated happily. But in integrator-deficient cells, our quality control machinery is absent. And so some polymerases can escape into the gene that haven't been properly matured, and these are now a little bit stupid, and they fall for this cryptic polyadenylation site and accidentally stop some of the times here in the middle of this intron. So now this intron can't get spliced out, and at some level, this intron, this retained intron, is exported. This gives it the opportunity to fold back on itself and then be recognized by PKR, which then activates an integrated stress response. And so we think that this is part of the longer term effects of integrator depletion. Okay, that's great, but everything I've just told you was in 293T cells, and you might be saying that's really not neurons, and so what are you really talking about here? And that's fair. And so what I want to tell you in the last 5 minutes is some work we were able to do with fibroblasts and cells that we obtained from these patients, from this cohort of patients. And what we had were 2 patients with mutations in integrator 8. And so we had 2 integrator 8 mutation samples and then 2 age-matched controls from this, again, this cohort in Holland. And these patients had insertions into the integrator 8 sequence that actually destabilized the protein. And because integrator 8 is really sort of fundamental for I guess the basal structure, the shoulder part of the integrator complex, the entire integrator complex was less stable, and we had reduced functionality. So again, some integrator activity, but a deficiency in integrator activity. So we grew up these fibroblasts, and we first just wanted to ask, now again, this is not our experimental system. These are fibroblasts from patients that have for long term had low integrator activity. We just wanted to ask, is deficiency in integrator function associated with an integrated stress response in these patient cells? So we first looked for ATF4. And again, here you can see that ATF4, this key regulator of the integrated stress response, is at much higher levels in the patients than the controls. We could also see phosphorylation of EIF2 alpha, again, suggesting this slowing of protein synthesis associated with this chronic or dysregulated ISR. And it really looked like this was also being funneled through this PKR because we saw this increased autophosphorylation. Now, I show this because also to show that fibroblasts are much prettier than 293 T cells. When Apoorva did this proximity ligation assay in the fibroblasts, again, looking for proximity between double-stranded RNA and PKR, he could see that the patient samples, both of them, were really full of these little yellow dots saying that there was double-stranded RNA being formed and recognized, and this was really absent in the control fibroblasts. Okay, so that's a little bit better, but it's still not neurons. Although if you're really waiting for like brain organoids, you're going to be disappointed. We're not there yet. But what we did do was take these fibroblasts and turn them into iPS cells. We here wasn't me, but was Ashley Schaefer. And what Ashley did, which was really wonderful, is she took the patient fibroblasts, turned them into iPS cells, and then in these sort of validated iPS cells, she corrected the mutation. So now we have otherwise isogenic cell lines that are really just different by 9 nucleotides or by the exact size of the mutation. And what we can see here, which is kind of remarkable, is when you repair correct the mutation, we bring back not just the normal levels of integrator 8, so we stabilize integrator 8, but we also stabilize the other subunits in this complex. So we also bring back integrator 1. Now we can look in these iPS cells at other sort of features of the integrated stress response. And for example, when we do Western blots for ATF4, we see that ATF4 levels are quite high in these iPS cells, as you might expect from the fibroblasts that gave rise to them. But we see that just correcting this mutation actually really turns down ATF4 now to very much basal, sort of normal threshold levels. In addition to this, we can also see that there's decreased phosphorylation of PKR in these corrected iPS cells. And so this was really exciting to us because it told us that in these systems where integrator had been deficient for an extended time, we were seeing the exact same sort of key features of a chronic ISR, a chronic stress response that Peter Walter had described in neurodegenerative disorders. And so that was very exciting to us. So again, I don't have brain organoids, but we were able to differentiate these into neuronal precursor cells. And here I'll just show you one of the experiments we did in these neuronal precursors from the patients or from the corrected control. And this is a staining with this J2 antibody, staining for double-stranded RNA. And so what you can see is in these neurons from the patients, or neural precursors from the patient's cells, there's really a lot of double-stranded RNA all through the cytoplasm. And again, just correcting this mutation really gets rid of this double-stranded RNA. And here I really want to thank Anastasia Kuzkina and from Vic Khurana's lab, who taught us how to take the iPS cells and grow them into neuronal precursors and now into neurons themselves. And so what I find sort of exciting about this is that now we can go back to this model that we built from a cell culture system that we can kind of beat the heck out of, having validated that in this more relevant cell culture model from these patient cells. And I think we can say that this integrated stress response and a sort of a chronic form of this, that we see in these patient cells is probably really contributing to this integrator-mediated disease. And this gives us a better handle on why loss of this quality control machinery is causing these problems. And I just wanted to notice, sort of where we're finishing here, that what is causing what we think, what we model, is causing the actual disease in these patients isn't something we would have ever seen if we just looked at something like RNA-seq, your standard differential expression analysis. Because many of these genes that we're discussing have more transcription in the beginning and less transcription in the end. So when you normally just count reads all over a gene, these balance themselves out, and the cell or the algorithm thinks that everything is good with these genes. And what our standard way of looking at RNA doesn't tell us is how many incomplete transcripts we're making, how many RNAs we start but we don't finish. And that's something that I think a concept that I think, as somebody who's been studying RNA for a really long time, we're trying to really now build into all of our analyses, is to make sure that we really estimate accurately full-length RNA expression. All right, so I just want to stop and go back to where we started, which is to say that, for many years, we sort of talked about what the function of pausing might be. And we said that we thought it was important to have a checkpoint early in elongation that would make sure that only mature and only the mature polymerases that have the proper repertoire of elongation factors and chromatin factors would be released into genes. And I think we now have, at long last, some evidence that that's really a key feature of regulation in our genomes, because we can find here that release of this immature polymerase, if you get rid of pausing, if you get rid of this quality control step, this can really cause trouble because of these immature RNA that are formed. So with that, I want to thank Apurva. I also want to thank very much Chen, who was a technician who worked with Apurva, Ryan, who helped us with a lot of the early IPS work, and Anastasia, again from Vic Khurana's lab, who helped us with the neurons and neuronal precursors. With that, I thank you all for your attention, and I'm happy to take any questions.
So, questions. I can start.
Okay.
So you mentioned that you have this neurodevelopmental disorder, but there are also cancer phenotypes. So is it known what happens when you have integrator mutations? What's the reason for the cancer phenotype? Is it also linked to this phenotype you described, or you think there might be something else?
Can you say that again?
Yeah, I mean, you mentioned that when you have mutations in integrator, you have various diseases.
So the question would be like, why various diseases?
If it also links to this mechanism you described.
I think so. I mean, and this is something that we're actively looking at, is chronic integrated stress response could potentially explain neurodevelopmental, neurodegenerative kind of defects. It could also chronic inflammation can also be related to some of the heart disease that's seen in integrator patients. For sure, ciliopathies and stuff are probably something a bit different, because that's often thought to be a difficulty in hedgehog signaling, for example. And cancers seem to be much more specific to mutations around where the phosphatase is recruited into the complex. So I don't want to say I think there's no subunit specificity to the different diseases, but I do think, and what we're trying to study now is how much of this is kind of integrator generically, and how much of this is a specific phosphatase defect or a specific signaling defect, specific pathway. And so we don't really know, but we do think that, so neurons have really, really long genes, right? And so this is one of these weird things about neurons, is that if you look at like cell type specific genes, neuronal genes are the longest. And so that's, again, one of our admittedly hand-wavy expressions for examples of why neurons might be particularly detrimental, this might be really bad in the neuron, is that those genes are more likely to both not be fully made, but also be, sort of making these, part-way transcripts that contain these retained introns.
Mm-hmm. Okay. More questions?
Thank you for the presentation. So I was wondering, the cells have mechanism how to prevent export of improperly spliced RNAs or unspliced RNAs. How come they are not stepping in here? Are they just flooded by the amount, or what's the reason?
Yeah, I think you hit the nail on the head, although again, this is still a little bit of, hand waving. But what we know, and I should have mentioned earlier, is that, I talked about, and I give as an example, promoter proximal pausing at protein coding genes, but that actually also happens at enhancer RNAs and long non-coding RNAs and upstream antisense RNAs, et cetera. So when you get rid of integrator, you actually increase the transcription and the length of eRNAs and link RNAs and everything. So the cell really does, a nucleus I should say, gets really pretty flooded with transcripts that are normally not made when you get rid of the integrator complex. And so the way that we're thinking about this, but this is something that's testable and that we're working on, is that the quality control machinery that would normally recognize these retained intron and these kind of aberrant transcripts, it's just overwhelmed.
Right.
And that's part of why some of this is sort of sneaking out. Now, I do want to note that even though quality control machinery is very good, at some level, a little bit of double-stranded RNA is kind of always, there's always a little bit. And so what I think we're seeing here is that there's a threshold that the cells tolerate and that we exceed that threshold, and so that we get this chronic signaling. And so I think this is a normally very good quality control pathway that just gets overwhelmed, and then we really exceed that threshold and in a sustained way.
Right, yeah, makes sense, thanks.
Okay, Ruben.
Congratulations, great data and very well presented lecture. My question would be, so what about the opposite? Are there any mutations which would actually stabilize the integrator and they would have, adverse phenotype or similar phenotype?
That would be amazing. I don't know of any. And so now that integrators kind of, I think, been better understood, there's more and more cohorts with integrator mutations that we're discovering. And we haven't yet molecularly characterized whether they're all loss of function or deficiencies, or, as you say, potentially like hyperactive integrator, right? So now nothing is getting transcribed properly because quality control goes crazy. The way that we think about it is that, at the promoter, there's kind of this race for who sees the pause polymerase first. If PTEF-B gets there first, the pause polymerase is happily released into the gene. But if integrator gets there first, then the polymerase gets kicked off and it has to try again. And so you could imagine really like severe issues with integrator overexpression. And so it would be interesting to see if something either caused the catalytic activity to be higher than it should be, or just the levels to be higher. And so we're trying to think of the best ways to sort of, in high throughput, molecularly characterize the different mutations that are coming in from the different cohorts around the world. Yeah.
And maybe a second question. So the overload with unprocessed mRNA, so wouldn't that also lead as a feedback to a genomic instability? So higher R-loop, etc.
Absolutely. And one of my colleagues, Fei Chen, has published a paper showing that over a course of days of acute loss of integrator, you do start to develop R-loops and you start to see genome instability. Sort of a really nice paper from David Cortez, who studies DNA and RNA conflicts in many situations, did a screen for things that lead to more replication transcription collisions, and integrator came up on his list. And so he got our cells from us and showed that even using our DTAG or our Degron cells, that he could show if you didn't have integrator, especially during that phase of the cell cycle where the polymerase is supposed to be kind of not in the way, that you get more DNA breaks if you can't keep the polymerase active and mobile. So I think there's probably a lot of things that happen in an acute loss of integrator that would be bad for cells and probably kill them. But since the patients live with a deficiency, we're trying to model that as well as continuing to understand what happens with the acute loss of it.
I also have to praise excellent lecture. Thank you very much. If I'm not mistaken, the integrator, at least the cleavage component, is also involved in the processing of snRNAs.
Yes, yes.
Could this incompetence of completing these long introns or the splicing be also a secondary effect of the defects in snRNA termination?
Yeah, so that was actually, I think, everybody's model. So when Ramin Shikatar first described the integrator complex, he described it as a unique machinery that created the ends of snRNAs, or led to the termination of snRNAs. In fact, the ends are made by a subsequent sort of chewing back, as you well know. And so when there were deficiencies found with integrator mutations, everyone anticipated that this was going to be an snRNA deficiency. And no one really could find differences in snRNA levels in integrator mutant patients. Or, we've looked so hard, splicing irregularities in integrator mutant patients are also very difficult to find. And so in a way, the human genetics was ahead of the molecular biology for a bit, because there were so many diseases in which integrator mutations were associated where splicing wasn't the issue. It was kind of telling us it's probably not just an snRNA regulator, but it took a while for us as a field to really appreciate that this was also something that was doing quality control and could cause premature termination at protein coding genes as well as enhancer RNAs, et cetera. So I don't want to say that there's no splicing defect, but we haven't seen disruption in even snRNA levels. And more recently, people like Steve West, who really study transcription termination, have reported that if you go through the area where integrator normally cuts the RNA, there's a perfectly fine polyadenylation signal or a T-tract where you can get DNA-dependent termination, and snRNAs are formed just fine because, as you know, the end is actually made by things like TOE1, and so even if you terminate a little bit farther downstream, as long as they're processed, you make okay RNAs. So we don't think the snRNAs are the main culprit here, but as you know, I love snRNAs, and so we keep looking at them because I guarantee there will be some situation in which that's actually the real problem.
Really nice. If I can return back to the integrator question. So has it been overexpressed? Because I'm just wondering what you mentioned, that there is a competition, because I'm just thinking if it's really quality control, that once it's very good, it just goes on, or if there is a really competition, because then it wouldn't be just quality control, but much more complex, right?
Yeah, no, this is so I do not know of anyone who succeeded to overexpress integrator. Because it's a 15 subunit complex, you have to kind of find the subunit or hope that there's a subunit or a couple that you can upregulate and then get the whole complex to be up. We have not succeeded in overexpressing integrator. My hope is that one of the mutants from some patient is going to be telling us, well, in this cell type, this mutant leads to an overexpression. So, we sort of tried to do it by RNA levels and say, well, this level is the lowest, so let's overexpress that one and hope the whole protein comes up. We are very bad at predicting biology, but mutants tell you when something interesting is happening, and so maybe some of these mutants will actually be a functional overexpression. But you're right, this tests a really important hypothesis about whether, quality control should be quality control. If everything's good, it shouldn't do anything, right? But if, in my model, it's more of a competition between PTEF-B and integrator, and you could have something such as an overactive termination complex that was actually, really suppressing gene expression. And that remains to be tested because we haven't really been able to overexpress it properly.
So if there are no more questions, we give a hand to Karen for the great talk.